So now we study the growing of the animal viruses inside the embryonated eggs. As we already know, there are the three different ways for growing of the viruses or growing of the animal viruses. Maybe in the laboratory, in the cell culture, then maybe inside the animals, and then in the embryonated eggs. So we will study them one by one, the growing the animal viruses in the embryonated eggs. There are different three ways the growing of the viruses. We can see the inside the body of the animals, the animal which is the specific host of that virus. For the study of that virus, we can use that animal and we can cultivate it in the most of the ways inside the animal, or maybe we can cultivate in the cell culture, just like we have seen the bacteriophages to cultivate to destroy the bacteria, and then we can see the embryonated eggs, how to cultivate the viruses inside the embryonated eggs. Why we cultivate the bacteria in the embryonated eggs? First of all, it's very convenient. It's very convenient to cultivate inside the embryonated eggs because we have the other ways, which are, if you compare them with the other ways, which is one the animals, it may be very expensive, and sometimes the same specific host of the animals, maybe we are not able to cultivate some of the viruses. We don't have the host in them, or sometimes we have the host, but we are not able to cultivate the virus inside that animals. So it's very it may be also very expensive if we need to buy the animals. If it is concerned with the guinea pigs, or it is concerned with the rats or the mouse or different ways, then it may be a little bit easy because we are already doing in the everyday's work. However, if the other animals, we need to use them for the big animal, just like cattle, or just like some of the viruses which need the primates. Of course, the humans viruses we need sometimes cultivate in the primates, so it will be very expensive and very inconvenient. And the embryonated eggs, they are host for the most of the viruses, so we can use them in very easy way. So what is the procedure? How to use it in the embryonated eggs? The hole is drilled inside first of all, the hole is drilled inside the shell of the eggs, which is very easy. Then the viral suspension is inoculated. How the viral suspension is inoculated, it depends on the routes. So there are different ways, there are different routes. Chorioallantoic membranes is there, amniotic membrane is there. So we will see them one by one, that which are routes and how it will be inoculated. First we see the egg shell, which is there. First of all, the shell membrane, the egg sac is there, which is very different, very prominent. And then there is the amniotic fluid, embryo, allantoic fluid, yolk, and the albumen. These are some different compartments of the embryonated eggs, which are used for the eggs to be inoculated. Now the viral growth, how the viral will grow inside the embryonated eggs. Of course, after the inoculation, the embryo will be died, just like we have already seen in the form of the bacteriophages, that after the infection of the virus, the bacteria will be died, and there will be only left will be the viruses. Similarly, in the form of the embryonated eggs, after the infection and after the injection of the embryonated eggs, the embryo will be died. But before the embryo died, it will be the virus has caused enough damage to the embryos, and it has left some inclusion bodies or lesions on the egg membrane, on the basis of which we will see that how we have already grown the viruses, and we can identify it, and we can isolate it, or we can use it for some other purposes. The purpose of the isolation and growth of the viruses, and then the growth for the vaccination, these are most important point of views for the embryonated eggs. Most of the times we are using for the isolation of different viruses, we are using the embryonated eggs. These viruses are used for the vaccination point of view because the embryonated eggs are host for most of the viruses. And for the vaccination point of view, we can just take the viruses from the field, and we can just cultivate them inside the embryonated egg, and then we can use some different tests to use them to see the vaccination point of view, to see the antigen, and then we can use it for the vaccines. There are different routes for the inoculations of the viruses inside the embryonated eggs. Most important of them are the chorioallantoic membrane inoculation, and then amniotic inoculation, allantoic cavity inoculation, and then the yolk sac inoculation. Just you see already seen in the last slides that what is the shape of the egg, embryonated egg. and we can inoculate the viruses in these cavities one by one. We will study them later in detail.